Um, all right. We have so far talked about carbohydrates as important biomolecules, proteins as important biomolecules. We are now going to lipids, which are also super important. So a few things about lipids. They are also made up of mainly of carbons, hydrogens, and oxygens, just like we saw in carbohydrates, but the ratio is really different. These are going to have these really kind of dense hydrocarbon tails of hydrogen and carbon. So lots more of hydrogens and carbons and less of the oxygens. Other big thing about lipids, or the main thing, like if something is a lipid for the purposes of our class, these are typically non-polar. We're going to see some special versions, like the phospholipids and stuff that have non-polar and polar qualities to them, amphipathic molecules. But in general, if you know something is a lipid, you can assume in general that it is non-polar. What does that mean in terms of its behavior when you put it into an aqueous solution?
It doesn't dissolve.
It's not going to dissolve. At least it's not going to dissolve without help. And again, a lot of the important signaling molecules in your body, like steroid hormones, are lipids, right? And so it means that you can't just have estrogen or testosterone flowing from one place to another in your bloodstream without help. So whenever we see something as a lipid, we know that we're going to have to get more complicated to actually move it around in this watery environment in your body. If you're trying to take cholesterol, which is an important part of a cell membrane, you can't just take it from one place and move it to another place. We're gonna see how we package it and how we actually manage to move it, in this case using like lipoproteins. So when something's a lipid, it's important to think about, okay, it's going to be more complicated to deal with in this aqueous environment, which pretty much everything in the body is located in. So let's talk about some of the core lipids you got to know. Triglycerides. So triglycerides are these are your basic, most of what you think of, like your fats and oils. They're named so it starts with a glycerol, which is like this little alcohol dude. With a C there. And 3 fatty acids. And fatty acids basically have like the acid is like a little carboxyl with these big hydrocarbon chains hanging off. Etc. This is where it might make more sense for me to just bring up an image from a PowerPoint rather than try to do it this way. So let me do that. PowerPoint, Silverthorne. Wait a second, hold on, sorry about this. Wow. What's going on here? Oh, there it is. Okay. Silverthorne, chapter 2. There it is. Okay, I'm going to share screen. And so these are what fatty acids look like here up on your screen. Yeah, if I take my pointer option, take the highlighter, this part here, this is like the little carboxyl. And then here you can just see this long hydrocarbon chain coming off of it. This one here, where every carbon has its full complement of hydrogens, this is as many hydrogens as you can slap on to this tail. This is what we call a saturated fatty acid. Saturated. I'm gonna just write. So saturated, it gets the name saturated because it's fully saturated with hydrogens. It has as many hydrogens as is possible to put on there. There's no more potential bonding ability left here to add anything more in terms of how many hydrogens are on there. However, if we look at, let me change the color here so it's a little darker. If we look here, or look here, these are also fatty acids. Again, we have our little carboxyl here. But if you look, there are these double bonds that show up in between the carbon tails. Whenever you have those double bonds, there, since you have one of the bonds of the carbon, the carbon is making a double bond, that means that's one less option for a hydrogen to bond there. So wherever you have those double bonds, those are called points of unsaturation. That's a place where you have one less hydrogen than like fully saturated, right? So this is a mono unsaturated fatty acid. If there's one point of unsaturation, one place where there's a double bond and therefore one less potential hydrogen for the full saturation of hydrogen. This one here has like 3 points of unsaturation. So there's even less hydrogens than this one. So this is this idea. These would be unsaturated fats. This is a mono unsaturated fat, poly unsaturated fat. One of the things that this picture does not show well is whenever there's a point of unsaturation, the tail gets a kink, physical shape kinks. So if I look at a fatty acid that's saturated, the tail is straight. If I looked at one of these guys that has 1 point of unsaturation, the tail would go, but where that point of unsaturation is, the tail would shift direction. If I have this dude here, it would go down wherever I have a point of unsaturation, I get a kink in the tail. So that is going to make a difference actually when we look at their physical behaviors and how they can stack up with each other and whether they're solid or liquid and stuff like that. So saturated fatty acids have a straight tail. Unsaturated fatty acids have a kink wherever there's a point of unsaturation. And then again, here we have, if we want to make a triglyceride, which is this one here, we have a glycerol and we add in 3 fatty acids. This is not showing it very well. All right, I'm gonna find something else here now. This is kind of showing here this idea of, this is the glycerol basically right here. Let's see, let's Slideshow. This is basically the glycerol. These are these, you know, they've been condensed in their formulas, but these are those hydrocarbon chains of the fatty acids coming off. So this is like your classic fat. Your classic fat is a glycerol, and then with these 3 fatty acids attached with their hydrocarbon chains coming off. So again, my kind of glycerol, and then my hydrocarbon chains. Back. So this is my classic triglyceride. If it is a saturated fat, these tails are straight. If it's an unsaturated fat, meaning that there's those fatty acids with those double bonds, then it tends to be a little more like this. The tails get kinked up. And saturated fats, these tend to be the ones we find in animals. These tend to be in plants, the unsaturated fats. You know, and fats are used for all sorts of stuff. Fats are used for storing energy. You can store like 2.5 times as much energy in a gram of fat as in a gram of carbohydrate. Fat is used to insulate. Fat is used as a core part of cell membranes. You know, fat is used for all sorts of stuff. Saturated fats in animals, because the tails are straight, they tend to stack on top of each other, and these tend to be solid at room temperature. So a saturated fat tends to be solid at room temperature. Like butter, or, you know, the fat in your steak or whatever. Unsaturated fats, because the tails are all kind of kinked up and these don't just layer on top of each other so tightly, these tend to be liquid at room temperature. Now, what do we call a fat that is liquid at room temperature?
Oil.
Oil. So these are also called oils. You know, and depending on how unsaturated, you know, if you have olive oil is monounsaturated fat. It only has 1 point of unsaturation. So it's a little kinked. It's still liquid at room temperature, but you put it in the refrigerator. I'm sure everybody has done this. You put olive oil in the refrigerator and it actually gets solid if you cool it down a little bit, and then you bring it back to room temperature and it's liquid again. Whereas polyunsaturated fats like safflower oil or corn oil, you know, those have lots of points of unsaturation, really kinked up tails. You put those in the fridge and they still stay liquid even as you cool them down. It's worth talking about hydrogenation, since that becomes an issue around just health issues around diet, with the whole thing with trans fats and things. So let's say I have some unsaturated fat. Again, this is going to be liquid. But let's say this is peanut oil. Let's say I have made I'm making peanut butter. I'm grinding up my peanuts to make peanut butter. And part of that process is expressing all that oil. But then I've got all of this liquid oil. So I let it settle, and all of a sudden the oil separates from the ground up peanuts. Like if you've ever had like totally natural peanut butter, it's like the oil and the peanuts separate, and you gotta like try to mix it up, and it's a pain in the butt, you know. And then instead you can buy Skippy or Jif, and it just stays creamy. The fats just stay solid and creamy, and you don't ever have to mix them up. It never separates. Like, how do they do this magic? You know, it's through the magic of hydrogenation. So hydrogenated fats, that means slapping hydrogen groups onto the tails. So adding in extra hydrogens, you know, so that is going to straighten the tails out more, and this is going to make this thing not as liquidy. So hydrogenation or hydrogenated fats are when you do this chemical process to modify these unsaturated fats to straighten out their tails, which is going to make them stay solid at room temperature, also makes them less likely to go rancid. Those points of unsaturation are places where it's more likely to break down. So they stay good longer, they stay creamy at room temperature. And it seemed like a great idea. And in fact, early, like when I was growing up, there was this big thing around margarine. You know, it's like, you know, there is a lot of evidence that diets with a lot of saturated fats are correlated with increased risk of cardiovascular disease. And unsaturated fats tend to be correlated with healthier outcomes. So people thought, why, instead of eating butter, let's take a plant oil, which is unsaturated, but then we'll hydrogenate it and turn it into something like butter. We'll call it margarine. And that'll be better for you than butter, because it's based on an unsaturated fat. And what they found was they made a huge miscalculation, because part of the hydrogenation process was creating what are called trans fats. So if you remember from chemistry, you can have the same molecule, you know, the same atoms, but you can have them have different conformations. I could have them bent this way, or I could have the same 4 molecules bent this way. This would be a cis conformation if these 2 things are bent to the same side. This is a trans conformation if they're bent to opposite sides. So trans fats are where you end up with these fats and they have kinks or bends in their tails that are not naturally occurring fatty acids. So basically you're creating, in this process of hydrogenation, they created fats that had bends in them that were not things that people would normally see just eating normal foods. And it turns out that our metabolism didn't really know what to do with them well, or it turns out eating trans fats, even though eating trans fats, you might think, oh, it's like eating just any other unsaturated fat. It turns out that the health outcomes were worse than eating hydrogenated fats. So making this thing that they thought was better for you and making margarine actually was actually worse for you than just if you'd stuck with butter, which was kind of ironic. Although at this point they have kind of worked things out. So now they can, you know, kind of hydrogenate things without making the trans fats, because now they're aware of this whole process and the problem in terms of health. But when you see the word trans fat,
Edgar,
yeah.
could you explain the little line, the black line that you made on top with the trans fat again?
Yeah, so again, the idea, we're going to see this a bunch. If I have some molecule, let's say these are my atoms. They can just all be carbons. Like, let's let them just be carbons. So I have 4 carbons in a row. And wherever there is a double bond, there's a kink. But I could actually have it go kind of like in a Z direction or more in a C direction. So if the bend makes it turn into a C, it's called a cis conformation. If it's more in the Z, it's called a trans conformation. So it's the exact same atoms with the exact same kinds of bonds, except one of them is flipped crossways and one of them are flipped on the same side. So physically, they take up space in a very different way. They're gonna bind to active sites on enzymes in a very different way. So enzymes in your, it turns out our body didn't have enzymes that could, easily work with these trans fats because they were not actually normal kinds of things you would get from eating diet through evolution of humanity. So, they ended up kind of stressing out your system. But yeah, we're going to talk about cis and trans in a number of different contexts when there's, some kind of a conformational change. Meaning a change in the shape of like a molecule. That's going to be at the core of how your rods and cones convert light into electrical signals for your brain. There's going to be these conformational changes where cisretinal becomes transretinal and starts a whole chain reaction in the photoreceptors in your retina. So yeah, this idea of cis and trans should feel familiar, and the idea that the exact same molecule can take on 2 different shapes. Again, we're going to see that in myosin. Like the myosin head can have the more cocked conformation or the kind of sprung conformation. You can have the same molecule in one shape, or snap it and now it's in this other shape, and snap it back and now it's back in the original shape, depending on the conditions. So those are triglycerides. Triglycerides are important. You should understand not hydrogen, that's more of a health thing. You should understand saturated fats, which are most of the fats in our body, or most animals. Unsaturated fats tend to be plant fats or oils. There are saturated plant fats like palm oil and things like that. So there's a few exceptions, but most plant fats are unsaturated. Morgan Rooks left. All right, so now let's talk about another really important thing we can do with this. Instead of adding 3 fatty acids onto the glycerol, we can instead add this phosphate containing polar group and make a phospholipid. So the phospholipids are going to be core players in our class. So let's talk about phospholipid. So again, there's that glycerol. And here we have our fatty acids. These are my kind of non-polar hydrocarbon tails there. But then we also are going to add in this polar head. So instead of just 3 hydrophobic things hanging off of it, we have this hydrophobic side, this nonpolar tails. But then we have this polar head, which is going to be hydrophilic. This is what we call an amphipathic molecule. Amphipathic means part of it wants to be, you know, like amphibians, they're like in the water and they're of the land. You know, this amphipathic molecule is of the aqueous solution and of the not the hydrophobic zone. So part of it wants to be just with the water because it's polar. Parts of it are going to organize away from the water because they're non-polar. This, you know, the main one that makes up your cell membrane is just called phosphatidylcholine or lecithin. You know, lecithin is just a phospholipid. This is the thing that makes up your cell membranes. And it's important to think about how this works. They self-assemble. You don't have to like bind them together or anything. If you just take some phospholipids, if you took a lecithin capsule from Woodlands Market and cracked it open, it'd be this greasy stuff in there, and you put it into water and you just shook it up hard, what would happen is each of these little things will have a polar side that's going to want to be towards the water. And a nonpolar tail that wants to kind of get away from the water. So they start making what we call the phospholipid bilayer. So the hydrophobic tails all kind of go in towards each other, kind of get away from the water. The polar heads all end up facing out towards where the water is. And again, this happens just spontaneously due to hydrophilic interactions. And it happens not just in one direction. It happens in three dimensions. So let me see if I want to find a nice picture of a phospholipid bilayer. Hold on a second. No, it's not that. Dagnabbit. Here we go. All right, I'm going to share. What? Wait a second. Just happened here. Oh, there it is. All right. So cell membranes, just as you can see right here in this kind of corner here. Where's my pointer options? This is kind of showing a phospholipid bilayer here. This is polar heads which are towards the water, all the hydrophobic tails, which all come to the inside. This is showing it in a more elaborate thing here. Here are all of the polar heads towards the outside, all the tails towards the inside. This is kind of showing it's like 3 dimensions here. Extracellular fluid, this is outside the cell. This would be like inside the cell. This is the basic structure of a cell membrane. And then there are also all of these proteins that are embedded in that cell membrane, which are going to play lots of important roles that we're going to be looking at over the course of this week. And then there is also cholesterol, like kind of stuck in between the tails. You can see on here, these little things kind of stuck in between the tails here are cholesterol molecules. So this is important. We're going to be spending so much of this class looking at what's happening at the cell membrane. You know, that's basically the boundary between the inside and the outside of the living things. We often call this the fluid mosaic model of the membrane. Fluid mosaic, because fluid is because things can shift, right? Things are only held together by the hydrophobic, hydrophilic interactions. One phospholipid can slide past another one. It never leaves the membrane because the tail wants to be in the middle and the head wants to face the water. But it would be like everybody's holding hands with everybody else, but you can let go of one person's hand and hold on to somebody else's hand. You know, so they're always kind of holding hands to keep the membrane intact, but which one they're next to at any one moment might be different from the next moment. So that's why it's fluid. Things are shifting. And the proteins as well. The proteins stick in there because of hydrophobic, hydrophilic interactions. And they can float around. They stay in the membrane because of the hydrophobic, hydrophilic interactions, but they can move, they can shift. So fluid, because things shift. Mosaic, because it's not just one thing. It's phospholipids, but it's also proteins and cholesterol and stuff like that. So we are gonna spend a lot of time elaborating on what is going on here at the membrane. Again, kind of making it clear, if I have my phospholipid membrane here, and the water is out here, my polar heads and hydrophobic tails. This holds together just because the heads are polar and want to be towards the water. The tails are non-polar and want to get away from the water, so they stay turned towards each other. The membrane proteins end up stuck in there. So this is some kind of membrane protein here. Remember the amino acids that make up proteins, those side chains can have different qualities. They could be polar or non-polar. So when you have a membrane protein like this, it's going to have the ends that are out are going to be polar. The ends that are towards the middle are going to be non-polar. So this thing wants to stay in place just because of its qualities. If you start pulling it out, this bottom part wants to be towards the water and this middle part would rather be in the hydrophobic region. You try pushing it down, it's the opposite. So this thing stays in place, not because it's bonded in there or anything, but by those same hydrophobic, hydrophilic interactions that just keeps the phospholipids assembled in their bilayer. Again, and that's why we have this fluid mosaic. This thing can actually float around and shift. It doesn't have to be locked into this particular phospholipid on either side of it. It can, as long as it's got the middle part inside the hydrophobic region and the poles facing it to the water, then it's just going to be able to kind of float around within the membrane. There are going to be ways to anchor them. We'll see using cytoskeletal components and stuff. Sometimes you do localize proteins on one part of a membrane or another, but they don't have to be localized. They can float around. In fact, it's going to be critical. We get into cell signaling in either this week or next. We're going to see very specific membrane proteins that have to float around from one place to another to do their job to facilitate signaling within the cell. The other important lipid are cholesterol and cholesterol-based molecules. Cholesterol is something that has it has rings. I don't even, maybe I can share it with you. Let me find a cholesterol. Okay, so I'm going to share. So this is cholesterol up here, which is a lipid. It's going to be a core part of cell membranes. It actually gets in between the tails. I'll talk about that in more detail in just a few moments. And then cholesterol is the starting point to make a whole bunch of steroid hormones. That's how you make cortisol, the main stress hormone. It's the base that you make estrogen and testosterone from. So it's important, like if you know a hormone is a steroid, that means it came from cholesterol, that means it's a lipid, that means it's hydrophobic. You know, so you need to be able to make those connections. Like if cortisol is if it's a steroid hormone made from cholesterol, it must be hydrophobic. And that's important when we talk about how it's going to be transported into the blood, for instance. Again, I could not tell you what a steroid ring system looked like, necessarily without looking it up. You don't need to know the details of cholesterol, but you should know that it is a lipid and hydrophobic. Doobie-doo. There are a few there's icosanoids, which are also lipids or lipid-based molecules. You spell it like this. These are like prostaglandins and things like that. Again, don't worry about the structure, but if you see this word, you can know it's a lipid and that means it's something about its chemical properties. Let's go back to the cell membrane though. So here's my phospholipid, or I'll just call it a cell membrane at this point, because now I'm really thinking about it in my cell. Here's the inside of the cell. Here's outside of the cell. Obviously, most is water. Talked about there is cholesterol that can be jammed inside between the tails. We'll talk about what it means to have more or less cholesterol jammed in there. So this is my cell. And there we go. So So I'm kind of trying to make this. This is like the cell membrane, and there's the inside and there's the outside of the cell. We really need to think right now at the beginning of the class about the properties of this membrane and how it affects permeability, allowing something to cross in or out of the cell. Because obviously it's going to be really important whether or not things can move in or out, or if they need help, or things we got to worry about that way. So let's talk about the permeability of the cell membrane. In fact, let me use my little type tool. We are going to talk about, make it a little bigger. So let's talk about what things can cross without help, what things need help. What kind of things do you think could go across the membrane without any help at all?
Elements like, like potassium or sodium or depending.
So it turns out that those are not going to be able to.
Okay.
Negatively charged
So
particles.
in general, things that are charged, so like sodium and ions, or even any kind of polar molecules. If you're a polar molecule, let me go just get the regular pen. There we go. If you're some polar molecule, you're going to want to dance with the water, with the polar solvent. So if you're some polar molecule and doop-de-doop, you're getting down here, you're like, no fun at all. This hydrophobic zone here has no charge. There's nothing interesting. You're going to go back up to where there is charge imbalance, and you're going to get to make hydrogen bonds and do that cool plus and minus dance with all the other molecules. So things that are polar tend to not cross the membrane. When they get up to the membrane, there is this, non-polar zone here, this hydrophobic zone. So polar molecules say, again, that is so boring, and hang out and stay with the charged stuff in the polar solvent in the water. So in general, polar molecules do not cross, without help. And we're going to see there are some exceptions, but in general, polar molecules do not cross without help. What about a non-polar molecule? And again, it has to get here, but let's assume it is on a carrier and it got up to the boundary here.
It will easily cross.
It'll just drift across. It'll end up on the other side. There's no reason it wants to stay connected up towards the water because it's not polar. It doesn't have any charge to attract it to this polar solvent. So non-polar molecules can cross freely. So this is important. Non-polar molecules will move across the membrane just because of their chemical properties, and they don't need anything extra. Polar molecules in general do not cross the membrane without help.
Could you please repeat that again? I'm so sorry.
It's just what it says here. Polar molecules do not cross without help.
Thank you.
And nonpolar molecules will cross freely, though. They don't need any help. If they get up to the membrane, they can just easily end up drifting to the other side. So in general, let me get back to here. Except, so it turns out if you have a polar solute that is really small, it tends to have this ability to just jam through before, except for very small polar solutes. So I'll show you how this works. So for instance, water. I have a water molecule. It gets down here normally from this argument I've been talking about. It doesn't want to go into the hydrophobic region, but it's really small. Its momentum, it has a certain momentum. It's moving super fast. By the time it gets in here and realizes I don't want to be here, it's already on the other side. So people talk about it kind of like a bullet or something. So really small polar molecules, even though it's polar, they tend to actually make it across. So that is the exception to this rule. Really small polar molecules like water. If water couldn't cross the membrane, osmosis would not be a major theme of our class, right? Obviously, water moves in and out of the cell, and we have to, that's why we worry about osmosis. But if this rule of thumb, polar things don't cross the membrane, then water wouldn't cross the membrane. So water in general does cross the membrane, and they think it's because it's so dinky. We're going to see other really dinky polar molecules that can cross membranes, like urea. When we do our lab next week, it will be very important that urea is a penetrating molecule. Even though it's polar, it's small and polar, and therefore can zip across. So very small polar solutes can cross. The cholesterol I was talking about in there, that tends to tighten up the tails and make them more and more packed against each other. So there are cells that basically don't let water even cross the membrane. And those are, in order to do that, you have to pack more and more cholesterol in order to keep the tails really tight. So there's no wiggle room there. But in general, in just your kind of run-of-the-mill cell, you assume that water can cross the membrane. And then getting back to, who was it? It was, I think it was Maria was mentioning ions. So ions are something we should talk about because they are a little non-intuitive. So here's my cell membrane. And here is, let's say this is an Na plus. So you might think, why it's polar, but it's really small. It says it's a single atom. So do you think a single ion can cross the cell membrane on its own without help?
Yes.
Right, so yeah, I mean, if you're first gonna think about it, it's like, yeah, it's dinky, sure. But it turns out that it can't. And it's because it's so highly charged, it anchors all these water molecules around it. So you start getting, they call it like a sphere of hydration. So the water molecules get into this really regular structure all around this thing because it's not just polar, it's like pure charge. So this is kind of like a snowball. Basically, it's like ice. Ice is just regularly highly structured crystalline water. So when you have an ion, it isn't just on its own. It's got its kind of ice ball that is pulled in around it. So effectively, it's actually big. So this thing actually will not cross. So won't cross without help. We're going to have all sorts of special ion channels that allow ions to cross in and out of cells, but there has to be some special channel in order for ions to come in or out of a cell. Because of this hydration, these water molecules that are pulled in really tightly around the ion, effectively it acts like a large polar molecule and therefore does not cross. So does that make sense?
Yes, Professor, thank you.
Sure.
Thank you so much.
All right, so that's that. I think the last thing we need to just briefly talk about nucleic acids, or the last of the kind of chemistry review. Luckily, there's not a whole lot we need to say about them. Obviously, y'all know RNA and DNA. You know, we're not going to look at them in lots of detail in this class. You should know the basics. DNA double-stranded, you know, with A's and T's and C's and G's. RNA single-stranded with A instead with U instead of T. RNA tends to be much more temporary. It breaks apart much easier. If you're doing lab work, just like, you know, oil from your finger will destroy RNA. RNA is definitely not like super stable. Whereas DNA, you go to the crime scene and get some blood off of, you know, the dagger that they found that's been lying under the floorboards, and they can find some intact DNA and try to figure out who the murderer was. So DNA is much more stable. Obviously, we'll talk more about a little bit more. In this class, we're not going to get a whole lot into the details of the DNA and the genome. We'll talk a little bit about it. I mean, the more and more we've been learning about DNA, the weirder and cooler things have gotten, just in terms of kind of long-term changes, all of the epigenetics, where, you know, the old model, like up to not that long ago, was that your DNA is given to you from your parents, and what version you have is the exact same version you got from them, and that's the version that you're going to be able to contribute to your offspring. And that experience that lays on top of that is for your life, but then the DNA that you give to your kids kind of goes back to the original blueprint. And now they're finding that that's not necessarily true. The whole, you know, if you've probably heard of idea of epigenetics. this is kind of long-term changes that happen to your DNA that actually persist in the germline. So this is often with methylation and things, these things that change aspects of the DNA, and then during all the DNA replication, they persist and go on to the germline cells. They've done some studies now with like rats, where they'll train a rat to be afraid of like the scent of geranium. You know, so it's not a natural thing that rats are going to be aversive to, but they'll train rats like geranium is paired with a bad experience, so they avoid the geranium. And then they'll breed the rats and look at 2 generations down, and the grand rats, you know, the grandchildren of those rats are going to still be avoidant of geranium smell. So there's like something that happened in that learning that actually did alter the DNA and is something that they passed on to their descendants. So this idea that your experience does not, you know, the old, you know, people used to make fun of Lamarck for saying like, oh, what an idiot. You know, he was saying like giraffes were passing on acquired characteristics. You know, that was so quaint and silly to think about now. But now we are realizing like, actually, there's something to it, that at least to some degree, we do seem to have the ability to pass on acquired characteristics. So this, again, we're not gonna, it's a whole other thing, but it's getting cooler and weirder than it ever was. And a little depressing, like trauma is something that you inherit from your grandparents, not just through the social interactions, but even within kind of the core wiring of your nervous system. So other nucleic acids we're going to see, obviously things based on the nucleotides. ATP is going to be a core player in this class. We've talked about it out the wazoo already. We've talked about some of this, one of the, you know, again, what's the job of ATP mainly?
Provide energy.
It's to deliver energy, to kind of take energy from one place to another to get things done. You know, the core process to make energy is going to be breaking down glucose in the mitochondria, but we're going to see it's this big, long, complicated process. You can't just do that all the time if you want energy right now. It's kind of like, you know, people talk about it being the currency of energy in the body. You know, that's kind of the little buzz phrase that people always use, but I think it's a good one. You know, if you think about money, it's like, you know, I have this skill, I can teach biology, and I do it here through the College of Marin, and they put money in my bank account, and then I can use that cash to go buy food or buy cool stuff off of Amazon, or go, like in the old days, go buy a ticket to go see a show when I used to be able to do stuff like that or whatever. So there's this instant way to transact using cash instead of this big barter thing. Like it would be a pain in the butt if I was gonna have to tutor the person at the grocery store and then they gave me some food in return, or, you know, so ATP, you do the kind of more complicated thing in the mitochondria, harvest the energy from the glucose, make the ATP, but then we have a billion ATP molecules floating around in every cell there to deliver energy, to pump ions or drive reactions that don't want to go on their own or whatever. So it's this way of having like just, it's like having a wallet with a wad full of cash. You're just ready to go and do what you need to do. You don't have to go through some complicated process to have energy on hand at the moment. So ATP, we're also gonna see, whoops. We talked about NADH and FADH2. These were these high energy electron carriers. These are going to be key players in cellular respiration, things, parts of the Krebs cycle, things going on inside the mitochondria to make ATP. Again, the basic idea with these things, for instance, NADH, you have NAD plus 2 high energy, plus 2 electrons, plus 2 protons, plus a proton, I should say, becomes NADH. This thing has high energy electrons, and when it breaks apart, it can donate those electrons to other things, which we then harvest the energy. Again, it's a lot like if I'm carrying some weight up high and then I drop it to a lower thing. As it drops, I can spin a paddle wheel or something and do something. So we're going to make NADHs, but then we'll use them in these redox reactions where you donate the electrons to something else that accepts them at a lower energy state. And in that process, we will be able to use that released energy to do interesting things that are going to be important for making ATP in the mitochondria. So we are going to see this later, either this week or next week. The other really important nucleotide-based things are some of the signaling molecules, cyclic AMP, cyclic GMP. So these are important signaling molecules. Inside the cell. Adenosine monophosphate or guanine monophosphate, right? Adenosine triphosphate with 3 Ps, adenosine diphosphate with 2 Ps, adenosine monophosphate with 1 phosphate. Cyclic AMP because it's hooked up in this ring form. These are going to be super important key players in signal transduction, like how you go from something binding a receptor to an actual action occurring within the cell. Core player in your rods and cones converting light into electrical signals. Core players in sexual arousal leading to an erection, all sorts of stuff. When we get into different drugs, like when we look at things like Viagra, it's working by affecting the processing of cyclic GMP inside the cells. So understanding these messenger cells is going to be important for understanding how things work, how things go wrong, how a lot of drugs have their actions. So when you see cyclic AMP, cyclic GMP, again, they're based on these nucleotides, either adenosine or guanine, and 1 phosphate and hooked up in this circle. Again, the exact shape doesn't matter. What matters if you see cyclic AMP or cyclic GMP is that you're thinking, oh, it's one of these nucleotide-based things that's an important signaling molecule inside the cell. So, again, we are going to come back and you'll spend lots of time with these dudes. All right, so let's see. So I'm going to wrap up with kind of combos. We just talked about carbohydrates and lipids and proteins and stuff, but a lot of the things are mixes. In fact, those membrane proteins that I was talking about, they always have carbohydrate groups attached onto the main thing is a protein, but there are also sugar groups attached onto that protein as well. And so if you have something that's mainly a protein but has some sugar groups on it, it's called a glycoprotein. Glycoprotein, glyco meaning sugar, so it's like a sugar-coated protein. Right, so it's mainly a protein, but it's got some carbohydrate groups attached. If you have something that is it's more carbohydrate with amino acid groups attached, peptide groups, you can have what's called a proteoglycan. So a glycoprotein is mainly protein with a bit of sugar on it. A proteoglycan is more of a sugar with a few of these peptide groups attached onto it. What do we call if you connect lipid groups onto a protein?
Lipoprotein?
We get lipoproteins. And so lipoproteins. These are going to be the key players in how we move fats around in the bloodstream. You know, these are going to be, there's going to be some protein part. There's a lipid part. I'm going to draw yellow because we usually think of fat as lipid. And then some protein part around the outside. So it's part protein, part lipid. The protein part will make kind of the outer interface with the water. So this is going to be the way we transport fats around in the bloodstream, by making a lipoprotein. You know, the lipid is kind of stored inside the core. The protein makes the casing around, which is actually polar and dissolves in the water. Depending on the ratio of lipid to protein, you can have high density lipoproteins, low density lipoproteins, HDLs, LDLs. We'll talk more about those when we get further on in the class. It's possible to have lipids where you attach sugar groups. So there's also glycolipids. Glycolipids would be like a phospholipid where you've added some carbohydrate groups onto it. So you can have lipids with sugar groups. So lipid, mainly lipid, but then add in some sugar groups. Just like a glycoprotein is mainly a protein adding on some sugar groups. So just kind of putting that out there. There is a whole variety of different things that work together. It's not always just a pure carbohydrate or protein or lipid. And I guess the other thing while we're talking about combos, so remember when we're talking about enzymes, and again, what kind of a molecule is an enzyme?
Protein.
Protein. But often an enzyme actually needs something in addition to just the protein structure to actually work. So sometimes they need these things called cofactors, coenzymes. So these are things that you usually get from your diet. Like a vitamin, when you think of what is a vitamin, vitamins are often just things you need to get from your diet that actually end up being they work together with the enzymes to help run their reactions. So sometimes, we'll talk about this in more detail later, but sometimes enzymes need an additional molecule in order to be fully functional. So that's when we talk about cofactors and coenzymes. So sometimes need these additional part to do their job.
Hi. I have a few questions about what you went over after ATP with the NADH and then the cyclic AMP and CAMP and CGMP. I'm just very confused.
What is your question about them?
Okay. So I understand that you're saying, I'm not quite sure where to start. Like I understand, I guess, with the last part, the cyclic AMP, the based on nucleotides and is important in signaling molecule in the cell, but I don't quite understand like what they are, how they work, other than.
We're going to talk more about their roles later.
Okay.
For right now, I just want you to know, if you see this, that it is, and again, it's adenosine monophosphate. It's an A with a phosphate, and it's hooked up in kind of a ring form.
Okay.
Right, just like ADP, it's adenosine diphosphate, ATP, adenosine triphosphate.
Okay.
So this is
So-
adenosine monophosphate. And rather than being important for energy, this one is important as a signaling molecule. It is a messenger.
Okay. Okay.
So
And
that
the CGMP is
is just another
Type.
similar. It's one of these. This, instead of the adenosine, it's got the guanine with
Guanine,
one
okay.
phosphate and hooked up in kind of a cyclical form.
okay. Got it. So it has a nucleotide that's attached to monophosphate, and
Or
it's
to a
important for
phosphate.
signaling instead of energy.
Correct. These
Okay.
are important for signaling.
Got it. Okay, that is very helpful. Thank you. And then going back to the NADH, so I just got a little confused. They use like high energy carriers, and then it sounds like when they break apart, they can deliver the electrons that are part of it to something else so that it like offers energy.
Right.
Is
Yeah,
that correct?
It
Okay.
would be like, here is like, I don't know, some tall bird holding like some ball. And here's some shorter bird that can catch the ball. And that there is a paddle wheel that gets hit as it drops, that spins around and does something interesting. So this would be like the NADH. This would be like the electrons falling down and getting caught at a lower level, and they do something useful with the energy that's released in that process.
Okay. Okay. That makes sense. I think I'm just getting caught up, which like I understand the concept of it when you explain it like that. I think like this, like when knowing what NADH, I think it gets confusing for me, like saying like NADH and then
You don't need to know. It's like, what is it? Nucleotide. It's, I forget what it actually stands for. Adenosine dinucleotide or something. You don't need to know the actual like structure of it. Just know that it's based on the nucleotide bases.
Okay, so same nucleotide bases, that one that has like a higher energy capacity when it gives its electrons to a lower energy.
Right. The NADH is a molecule that's that's in this family.
Okay.
That can, when it's in this NADH form, it has electrons that are held in a very high energy conformation, or they have a lot of energy, and when you do a redox reaction and give them to some other molecule that holds them at a lower energy, you can capture that energy and do something interesting.
Okay. Just writing down. Okay.
Again, the best metaphor I can think, or simile or something, is like if you have a bunch of water up in a reservoir up at the top of the mountain, and then you let the water run down the hill to a reservoir at the bottom of the mountain, and then stick a paddle wheel in the stream.
Right.
So it's like that.
And it's not clear yet, like what that paddle, where it will give its energy, just knowing that it can
Correct.
capture.
Yeah, we're
it
going to
and
talk
do
about the cytochrome, the electron transport chain in the inner membrane of the mitochondrion and all that. But
Okay.
right now,
For now.
that's to be, yeah, that's spoilers.
Okay. Okay, great. Thank you. That is really helpful. And then the last question I have is just so that I know I got this wrong on my, when we went over it last week, I understood it with osmolarity of when you're saying that NaCl breaks apart, it'll have double because it completely dissolves versus glucose dissolves, but it doesn't dissociate so that it has a lower osmolarity. But then when you went into
Or
this,
it's I mean, glucose has identical osmolarity as to molarity.
sorry, say that one more time.
The osmolarity and the molarity of glucose will be identical.
Got it.
Because,
But the
because it-
NaCl osmolarity is twice as much as the molarity.
correct.
And that's this, and then the CaCl2, because it will break up, dissociate into 3 molecules, right? Then it would be 3 times the molarity because it has the different.
Correct.
Okay. Just want to make sure I understood. I know I got that wrong. Okay. Thank you very much.